So we have now the results of the screening for different kinds of mutations, or another way to look at that is yeast cells that are deficient in some cellular process. So this is sort of like a phenotype for a single-celled organism. The question is, can it grow as well as a wild type, or is it maybe missing something that would make it grow differently? So we started off with cells that were genetically uniform. We exposed them to ultraviolet light, and so therefore any differences, any significant differences in the phenotype or the way they grow would have to be due to a genetic change, because they've otherwise been exactly the same up to that point. So I'll just go ahead and film one video that shows you all the different tests that we did and the results of those. So over here, we're going to go right to left. This is the temperature sensitivity. So these plates were growing at 37 degrees Celsius. This is the YPG glycerol plate, G for glycerol, forcing those cells to do aerobic respiration. This was our DNA damage assay. And over here we have a stack of oxidative stress plates. So I'll show you the results and talk about what they mean all together in this video. So starting off with the temperature sensitivity for this one, if you remember, we did the streaking. So we streaked the cells trying to get individual colonies. These grew up a little bit more because they were at that high temperature. Probably gave them a little bit longer to grow than maybe I should have. Again, if you remember, we're going to compare the sizes of the colonies. So over here we have the wild type, and those colonies are maybe on the small side. I guess I should give you a little caveat here, though, that yeast cells growing close to each other are going to exhaust the nutrients that are available in their vicinity. So this wild type streaking over here was a lot more cells than, say, across diagonally the B suspected mutant. These cells had more room and therefore more nutrients to grow, take advantage of. At any rate, we're trying to find, or we're looking for cells that would grow more poorly under this temperature sensitivity. Not to say that mutation couldn't make cells grow better. That is definitely a possibility. Could be that they've sort of sacrificed some kind of checkpoint and decided to grow faster. Not decided, but say, as a result of some kind of genetic change, they would grow faster under high temperature. That is possible. Always good to keep these things in mind when you're looking for phenotypes. Really ideally, if we had total temperature sensitivity, this would be a condition that would prevent the cells from growing really at all, or certainly making a marked difference in how they grow compared to the wild type. Don't really see that here. What I think I see is really just differences in density. So the cells that are growing closer together grow into smaller colonies, and the cells that grow farther apart, more isolated, they can grow into bigger colonies. So I'd say maybe nothing really remarkable to report there. These are the YP glycerol plates, and altogether there was a little bit more difficulty growing. I probably got these out of the incubator a little bit early. We could give them more time and see how big those colonies grow up. But at this stage we can more or less compare them. I'm a little bit surprised with how these turned out. They're different colony sizes, even though the colonies can be spaced equally. So there's sort of a range of colony sizes, even for the wild type. And so comparing those across the suspected mutants, everybody's got pretty small colonies. Some of these are a little bit interesting, maybe something you'd want to get in and test a little bit further, but say F here has the dense streak there, and then a pretty sparse streaking in this area. Same for G, pretty sparse out in this area. Could be the result of differences in streaking, could be that cells were put in here that didn't grow up. So as with a lot of things we do in introductory labs like this is giving you a flavor for what you would look for. If you were in a research lab and you were really interested in isolating these aerobic respiration mutants, you would certainly do more testing after this point. But kind of at first blush, we might have wild type compared to F, wild type compared to G, maybe a little bit of a difference there. We could run subsequent tests, and certainly replication would help us to confirm if that was the case, that they grew differently. Moving on to our DNA damage assay. If you remember, we had a dilution, so diluting from the full strength solution, 1/10 and 1/100th strength. Zoom in a little bit. This was the error that we made and acknowledged last week. So wild type full strength, and then these were all full strength. And what I meant to do was 1 to 10 and 1 to 100, and those got put over in this area. So kind of ignore these. Ignore those pieces. But kind of good, I think, to remember that we did kind of overcome a problem by just dotting them off to the side. Okay, so what we want to compare here is we want to compare how A, B, and C grow compared to wild type. And probably the best dilution for us here is this middle one, probably for everybody. The middle one, you can still see some light coming through. The most dilute, the 1 to 100 dilution, is just, I'd say, too few colonies to really notice. And now we want to compare. So if we've established that's the right dilution to compare, 1 to 10, we're going to compare up on the top. This is the ones that were no UV, and middle was 100 microjoules per square centimeter, and the bottom was 200. And so we would notice if there was a DNA repair deficiency, the cells that were exposed to the UV light would maybe drop off in density compared to the ones that were not exposed. So roughly the same here, 0, 100, 200. Roughly the same, 0, 100, 200. Certainly wild type, 0, 100, 200. So looks like maybe nothing to report there. Same on this plate for the D, E, F, G mutants. Comparing the middle dot, same, 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 same. This is actually a pretty robust way to compare cell performance or cell growth. So if you put roughly the same number of cells on there, you would be able to see them as being dense or less dense as they grow up, given they had the same amount of time to grow up. All right, so I didn't see a big difference on any of those. Let's look on to our oxidative stress test. So if you remember, we dotted a disc that was exposed or soaked in water or hydrogen peroxide, and we put it onto a plate that had just a uniform streaking of cells on there. And sure enough, we see this halo effect around the hydrogen peroxide. So that's just what we expected to find. I'm going to roughly dot the region where those cells stop growing. So this is the zone of inhibition, or basically the distance from that circle, the dot of paper, the distance from that cells really weren't able to grow during this time. So they were able to grow right up to the circle in the water. That's no surprise. Water shouldn't be a problem for yeast cells to grow next to. But hydrogen peroxide is an oxidative stressor, and so they would have a distance that they can't really grow across. So for this one, we're going to measure the diameter. So I'll put my ruler down on there, and I'll give that 3.7 centimeter diameter. And you can see it's basically a perfect circle. If you wanted to, you could take multiple measurements across that and compare them. Let me try to do this a little bit faster for these other ones. So through the center all the way from one point to the next, 2.9 centimeters diameter. Pretty good circle on here. I don't know if I trust it right up next to the edge of the petri dish, so I'm going to avoid that area if I can. Give that a 3.4 maybe. All right, so as I'm doing this, think to yourself what it would look like if some of these cells were more or less sensitive to oxidative stress. 3.8. 2.7. Hmm. 3.6. Two to go. .4. And finally, I don't know why it seems to drop away next to the edge of the dish. Like I said, I don't know if I trust that. So I'm going to focus across the middle. 3.5. Okay, so we would put these into a data table. 3.5, 3.6. So we have a range from 3.8 was our maximum, 3.7 for the wild type, and then all the way down to 2.7 on this mutant, suspected mutant D. So if you thought about what we'd be looking for, we would probably expect ourselves to be more susceptible to oxidative stress, and that would look like a bigger zone of inhibition. So 2.7 here is smaller than the wild type. So what's going on? Well, we could have generated a mutant that is more resistant to oxidative stress. What I think probably is more realistic is that I was perhaps a little inexact with the amount of hydrogen peroxide that was on that disc. So soaking it and then drawing it up the side of the petri dish, it's possible that more or less of that hydrogen peroxide was contained on the disc, and that might have caused the effect. So it might be that I gave the wild type one a heavy dose of hydrogen peroxide, a larger volume would have caused a higher concentration, and these closer to A and D here. Oh, that's interesting. I think I did that one backwards. So, messed up my labels on that one. I think this is probably correct. So 2.7, that one's weird in a few different ways. So nothing that we see that's more concentrated. Certainly if we wanted to, again, confirm that this was the case, that we had a difference, a significant difference in oxidative stress response, we would want to run statistical tests, and statistical tests require replication. So we would need to do the same test over a few replicates of the same wild type and the same suspected mutants, and see if these numbers are continuously or consistently different between the wild type and the suspected mutants. So we're giving you a little flavor for what you can look for. I don't know if we've shown a very strong evidence for the results being that different across these suspected mutants, but it gives you something we could look into investigating more in the future.